My name is Jessica Lopez, and I'm one of the genetic counselors here at Greenwood Genetic Center. Your provider has recommended a gene panel test, and I'm here to provide you just a little more information about that. First, let's review some background genetic concepts. Our bodies are made of millions of cells. Each cell contains a full set of our genetic instructions, which are organized into structures called chromosomes. If we stretch out the chromosomes, we begin to see the genetic code written out in letters. These letters make up thousands of individual instructions called genes. Genes code for proteins that help our bodies grow, develop, and function. If you think about the genes made up of letters providing instructions to the body, you can imagine that it is important that the instructions are written clearly in a way the body can understand. This test specifically looks at those instructions and how they're spelled. There are many different types of spelling changes that can occur within the genes that then cause problems for the way the body forms or functions. These changes have historically been called mutations, but are now more commonly referred to as variants. When we use genetic testing to look at the spelling of genes, we can do that by looking at one specific gene or by looking at multiple genes at the same time. Looking at multiple genes that cause similar clinical features within the same test is referred to as a panel. Some panels have already been developed by the lab, like in the case of genes associated with syndromic forms of autism or connective tissue disorders. Other times, a provider may recommend a custom or focused gene panel that is more specific to a patient's symptoms. If available, your provider may recommend that we get samples not just from the patient, but also from the patient's parents. Including parent samples may help us understand a patient's test results. If one or both parent samples are not available, we can still run the test for the patient alone. There are 3 different results that we can get from this testing. The first is a negative or normal result. This means that we looked through the genes included on the panel and didn't find any changes that we think are causing problems. We can also find a positive or an abnormal result, which just means that we found a change in one of the genes that we think is causing part or all of a patient's symptoms. The third result is a variant of uncertain significance, or VUS. This means that we found a spelling change in a gene, but we aren't quite sure if the spelling change causes a problem or not. In these situations, your provider might recommend additional testing or evaluations, or they might wait until they've learned more information about the change. As we learn more information, these variants of uncertain significance can be either upgraded to pathogenic or disease-causing, or downgraded to normal results. Please know that in genetics, we are learning new things all the time. So even if you receive a normal result now, it's possible that we will contact you in the future with an update. Patients who have had a panel test done through the Greenwood Genetic Center laboratory have the option of adding on a quick analysis if the results of the panel are non-diagnostic. QUIC stands for Quickly Uncovering Important Clinical Knowledge. All of the gene panels at GGC are performed on either an exome or genome backbone, meaning that data from essentially every gene is generated. A quick analysis is a brief look at the remainder of the genes outside of the genes that were included on the panel to continue to look for harmful misspellings that could be related to a patient's concerns. If requested by the patient, a quick analysis can be performed at the same time as the panel or after the testing is completed. It's up to you whether or not we perform this additional step as part of the patient's test. The intention of this testing is to identify a cause for a patient's concerns. However, we can sometimes uncover unexpected or incidental information that can impact the patient and/or their family members. For example, the test may reveal that a patient may develop additional health problems in the future. If parent samples are included, it may reveal that a patient's parent has the same genetic condition as their child, that parents are related to each other, or even that one of a patient's parents is not their biological parent. It's important to keep in mind that this test doesn't detect all possible genetic changes, so there might be more testing recommended if this test comes back normal. Results of this test will be available about 8 weeks or 2 months after samples are received by our lab. Your GGC provider will contact you to review these results and next steps. If you have any questions about this testing, reach out to your Greenwood Genetic Center providers and we'll be happy to answer your questions.